Unraveling the Mysteries of DNA Sequencing, Techniques and Technologies. Title: Unraveling the Mysteries of DNA Sequencing, Techniques and Technologies. Introduction. DNA sequencing has revolutionized the field of molecular biology, providing scientists with valuable insights into the code of life. It allows us to decipher the genetic information stored in DNA, unlocking a world of possibilities in healthcare, agriculture, and beyond. In this article, we will explore the techniques and technologies behind DNA sequencing, shedding light on how this remarkable process works. Understanding the basics of DNA sequencing. DNA, or deoxyribonucleic acid, is the blueprint of life. It contains the instructions for building and maintaining an organism. The breakthrough discoveries of James Watson and Francis Crick in the 1950s paved the way for our understanding of DNA's double helical structure. However, it was not until the 1970s that the concept of DNA sequencing was introduced. DNA sequencing is the process of determining the precise order of the four nucleotide bases, adenine, A, cytosine, C, guanine, G, and thymine, T, in a DNA molecule. This information is crucial for understanding genetic variations, identifying disease-causing mutations, and tracing evolutionary relationships. Sanger sequencing, the gold standard. First developed by Frederick Sanger in the late 1970s, Sanger sequencing quickly became the gold standard in DNA sequencing. It relies on a clever mechanism that involves DNA replication, dideoxynucleotides, eotides, ddNTPS, and gel electrophoresis. In Sanger sequencing, DNA is replicated in the presence of both normal nucleotides, dNTPS, and a small amount of ddNTPS, which lack the 3-hydroxyl group. The introduction of ddNTPS results in chain termination at different positions, generating a set of DNA fragments of varying lengths. These fragments are then separated using gel electrophoresis, creating a sequence ladder. By analyzing the ladder, experts can determine the order of the nucleotides and unravel the DNA sequence. Next-generation sequencing, NGS, technologies. While Sanger sequencing has been instrumental in many scientific breakthroughs, it is not without limitations. It is time-consuming, expensive, and limited in its ability to sequence large amounts of DNA. The development of next-generation sequencing, NGS, technologies address these drawbacks and open new doors in genomics research. NGS works by simultaneously sequencing millions of short DNA fragments, creating a massive amount of data in a single experiment. Several techniques fall under the umbrella of NGS, including pyroseq, ENSing, ion semiconductor sequencing, and sequencing by synthesis. Pyrosequencing, developed by 454 Life Sciences, detects the release of pyrophosphate molecules during DNA synthesis. By incorporating nucleotides one at a time, researchers can measure the light emit as pyrophosphate is released, contributing to the DNA sequence. Ion semiconductor sequencing exploits the ionization that occurs when nucleotides are incorporated into a growing DNA chain. As each nucleotide is added, hydrogen ions are released, and a semiconductor or chip detects the resulting pH change, thus revealing the DNA sequence. Sequencing by synthesis, as seen in Illumina platforms, capitalizes on fluorescent nucleotides to sequence DNA. Each fluorescently labeled nucleotide emits a different wavelength of light upon incorporation into the growing strand. Through the detection of these emitted lights, scientists reconstruct the DNA sequence. Advancements in NGS technologies. Since their inception, NGS technologies have undergone rapid advancements, resulting in increased sequencing speed, reduced costs, and improved accuracy. These advancements have enabled large-scale genome sequencing projects and facilitated the exploration of complex genetic variations. Furthermore, NGS has paved the way for applications such as metagenomics, epigenomics, and transcriptomics. Metagenomics allows scientists to study complex microbial communities, while epigenomics focuses on the modifications that influence gene expression. Transcriptomics provides a snapshot of gene activity, aiding our understanding of diseases, development, and cell function. Emerging technologies. As technology continues to evolve, new sequencing methods are emerging. One such promising technique is single-molecule sequencing, also known as third-generation sequencing. This cutting-edge method aims to sequence individual DNA molecules without amplification or cloning. Another approach, dubbed single-molecule real-time sequencing, captures the incorporation of fluorescently labeled nucleotides in real time, allowing for direct observation of DNA synthesis. In the quest to unravel the mysteries of DNA sequencing, researchers have made significant strides in understanding the complex processes that govern the code of life. DNA sequencing techniques and technologies have played a crucial role in this endeavor, enabling us to obtain detailed genetic information with remarkable precision. This article will delve deeper into the intricacies of DNA sequencing, shedding light on the various methods employed and the technological breakthroughs that have driven the field forward. One of the foundational techniques in DNA sequencing is known as Sanger sequencing, developed by Frederick Sanger in the late 1970s. This method quickly became the gold standard, relying on a clever mechanism involving DNA replication, dideoxynucleotides, ddNTPS, and gel electrophoresis. In Sanger sequencing, DNA is replicated in the presence of both normal nucleotides, dNTPS, and a small amount of ddNTPS, which lack the 3-hydroxyl group. The introduction of ddNTPS leads to chain termination at different positions, generating a set of DNA fragments of varying lengths. These fragments are then separated using gel electrophoresis, creating a sequence ladder that can be deciphered to determine the precise order of the nucleotides in the DNA molecule. While Sanger sequencing revolutionized the field of genomics, it also had limitations. It was time-consuming, expensive, and not suitable for large-scale sequencing projects. Therefore, researchers ought to develop alternative approaches that would overcome these obstacles and pave the way for more efficient and cost-effective DNA sequencing. This led to the emergence of next-generation sequencing NGS technologies. NGS technologies encompass a range of techniques that enable the simultaneous sequencing of millions of short DNA fragments, generating a massive amount of data in a single experiment. Among the different NGS methods, pyrosequencing, ion semiconductor sequencing, and sequencing by synthesis have emerged as prominent approaches. Pyrosequencing, developed by 454 Life Sciences, detects the release of pyrophosphate molecules during DNA synthesis. By incorporating nucleotides one at a time, researchers can measure the light emit. As pyrophosphate is released, thereby contributing to the reconstruction of the DNA sequence. Ion semiconductor sequencing, on the other hand, exploits the ionization that occurs when nucleotides are incorporated into a growing DNA chain. Each nucleotide addition releases hydrogen ions, and a semiconductor chip detects the resulting pH change, providing information about the DNA sequence. Say, quencing by synthesis, as seen in Illumina platforms, utilizes fluorescently labeled nucleotides that emit different wavelengths of light when incorporated into the growing DNA strand. By detecting HESA-emitted lights, scientists can reconstruct the DNA sequence accurately. Advancements in NGS technologies have been remarkable, driving increased sequencing speed, reduced costs, and improved accuracy. These advancements have facilitated large-scale genome sequencing projects, enabling researchers to explore complex genetic variations and further their understanding of human genetics and diseases. Moreover, NGS technologies have broadened the scope of genomics research. With applications such as metagenomics, epigenomics, and transcriptomics. Metagenomics allows scientists to study microbial communities and their interactions, while epigenomics focuses on the modifications that influence gene expression. Transcriptomics provides insights into gene activity, shedding light on diseases, development processes, and the functioning of cells. As technology continues to evolve, new sequencing methods are emerging at a rapid pace. One such promising technique is single-molecule sequencing, also known as third-generation sequencing. This cutting-edge method aims to sequence individual DNA molecules directly, without the need for amplification or cloning.